Hi, I'm David Carlson, and I'm an assistant professor at Duke University. I'm going to be talking today about learning electrical connectomes of neuropsychiatric disorders and behaviors. I wanted to begin by framing the research goal. I'm particularly interested in understanding networks of the brain and how they relate to behaviors or neuropsychiatric conditions. My lab is developing explainable artificial intelligence techniques that learn networks of neural activity that are biomarkers for several conditions. These techniques need to explain themselves to communicate with domain experts. And one of the things that I'm really passionate about is moving beyond just the statistical analysis and actually using the artificial intelligence tools as a partner in experimental design. So we've already used our machine learning methods to learn testable hypotheses from data, and we've partnered with the Collective for Neuropsychiatric Engineering led by Kafui Daraza to design and run experiment on these hypotheses. So moving forward, I'm first going to highlight some of the properties of my machine learning approaches and models, and then I'll explain a study that we did in partnership with Kafui Daraza, where we actually use these explainable AI tools to derive hypotheses and actually design confirmatory experiments. So I wanted to begin by just setting up how we're viewing our emotions here and how we're going to be viewing emotions as coming from different latent networks of the brain. For example, you can have a fear emotion, and we're going to assume that that is going to come from a latent network corresponding to fear. When you experience fear, that means that there's a biological neural network that is activated and producing that emotion. This idea has been in the literature before, and it's been explored several times in imaging studies of the brain. For example, in this Craigel et al. Study, they asked humans to self-report emotions, and they used fMRI to decode emotions with fairly high accuracy. The key result for us here is that there are clearly different neural activations under each emotional condition. So different part of the brain is activated and communicating under each emotional condition here. And you can see that on the left here, where you can see some of the different emotional setups. So for example, contentment is in red, and you can see the part of the brain that is activated when you are content. And you get different parts of the brain that are activated under these different emotions. And so this is kind of the basis that we're thinking about here, that emotions really are latent networks. In our studies, we're going to be interested in network representations of different emotions or conditions in animal models. And we're really interested in animal models because we're going to be able to do a lot more interventions there. Unlike humans, animals cannot self-report. So we're left with this challenge that we cannot differentiate between mouse emotions simply by looking at them or by asking them. This leads to a challenge that we need to find some way of identifying networks that correspond to our emotions or neuropsychiatric risk. So we're looking at this. We have this problem where we need to think about how we can use behaviors and training data in order to help guide us to what we're looking at in our emotions. The other thing that we're going to be looking at is we're going to be looking at what's called local field potentials. In particular, we're interested in fairly fast and temporally precise network properties. Right, so these local field potentials are going to come from implanted electrodes, and these electrodes are going to be all over the brain. And each electrode is going to capture the low frequency pattern. When we're looking at a low frequency pattern inside the brain in a particular brain region, we're going to call this a local field potential. This is very highly related to the EEG that you can record outside the brain. It looks similar, but it's a lot more spatially precise, and it's a lot higher fidelity. And in addition, we also get high frequency information, which you can use to calculate things like action potentials from different neurons, which I'm going to ignore for now and focus instead on the LFPs. So what I'm showing you here on the left is I'm showing you for each of the different brain regions that was implanted, I'm showing you a local field potential trace. Now, this local field potential trace, we're assuming that there's one per brain region. If we have multiple wires in a brain region, we're just going to average them to get a single local field potential. And we're going to do this for reliability. This loses a little bit of information, but we're really interested in networks that hold up across individuals or across animals. And so in that case, we think that the least resolvable unit is a brain region. And that's where we can get consistency and reliable information that is the same across these different individuals. So LFPs have been used kind of historically in a lot of different ways. They're believed to reflect the sum of activity from many neurons within a brain region. And people have used them to say, take them and break them down into different brain waves. So for example, you might have your theta and your beta and your delta. And you have different types of brainwaves here that represent different properties of the neurons, right? So if we think about the fact that we have these local field potentials, if we come up with a network over local field potentials, we're going to believe that they are going to reflect broad properties of neurons. So what we need to do is we need to figure out some way of capturing the important information of the local field potential. People historically have used these power features in different frequency bands. Now, for our analysis, because we're interested in networks of the brain, we're really interested in saying when these brain regions are communicating. So we really need to capture properties that give us this communication. So in particular, we're going to need to capture it in different frequency bands. So what I'm showing you here is I'm showing you the hippocampus, and I'm showing you just filtered to a particular frequency band, and I'm showing you the prefrontal cortex filtered to a particular frequency band. Now, one of the things that people have looked at historically is just whether they have similar patterns, right? So if the hippocampus and the prefrontal cortex have similar patterns, this means that they are functionally connected in some sense, that they are communicating in some manner. That might be direct communication, it might be a non-direct communication. For example, you have something upstream that's sending them both the same information. And this is a measure of communication. And one of the things that we really want in our network is not just whether two brain regions are correlated, but actually, are these two brain regions sending information to one another in this frequency band? So we can think about that by coming up with these measures of directionality. So we can think about what it would mean for the hippocampus to be sending information to the prefrontal cortex. So we take our hippocampus here, and we say it's sending information to the prefrontal cortex. We think that if there's a temporal lag, and then the peaks kind of match up really cleanly with what's going on in the hippocampus, then we think the hippocampus is sending information to the prefrontal cortex. Now, if we look at the setup here, we think that this is matching a little bit, but not exactly that perfect, whereas if we look at the alternative relationship, it actually matches up a little bit better, where this is the idea where we might be sending information from the prefrontal cortex to the hippocampus. Now, I'm sketching this out here just by drawing lines and showing you that these peaks are lining up in interesting ways and that information might be flowing from one direction to the other based on temporal lags. So when we're defining mathematical networks, we're not going to be using these kind of conceptual plots. We're going to formalize this based off something called Granger causality. But really the key point here is that we believe our networks need to capture directional flow of information. And we think this is really important for when we start to go to develop our hypotheses of how these networks are actually working. So the way we actually go about doing this is by defining our networks. And I want to start by showing you what a network actually looks like in our analysis. This is a network related to stress. Because we are modeling the electrical signals of the brain, we call this network the electrical connectome or electome network. The electome captures the spectral or frequency properties of the brain, including the power coherence and directionality properties. So on the bottom left, we have a circle plot. And each brain region is represented by a different wedge. So for example, up at the top left of the circle plot, we have VTA, or the ventral tegmental area. Each brain region has in there these numbers that say 10, 20, 30, 40, 50. This is the frequency in hertz. So we're breaking it down into frequency band. A mark on the outside of the circle plot indicates that power is much higher than background levels in the brain when this network is active. So we think that when this network is active, there's power at about the 8 hertz or 2 to 8 hertz band in the ventral tegmental area. If there are lines connecting to brain regions, we say that this is coherence, that they are communicating in some manner. So you can see here that we have a lot of lines connecting all the brain regions in this particular network. This network, in the brain regions that were measured, we kind of get communication everywhere. That's not always the case. And we really see a lot of lines, and we've colored the lines here blue and magenta. And this really is just to help us know that there are 2 frequency bands that were picked up by this electome network. So the blue band here is at the 2 to 8 hertz frequency band, and the magenta is at the 12 to 20 hertz frequency band. So not only can we view which regions are involved in this network, we really want to use that idea of this directionality to think about how information may be flowing in the network. So when we think about the electome network, we have these frequency patterns, and then we can go over here to the right and say, well, what are the leading regions? Where do we think this signal is actually originating in this network? Well, we think this is in the 2 to 8 hertz frequency band in blue. We believe that the primary leaders of this are IL and the prelimbic cortex, which you can see from the arrows beneath it. And in the 12 to 20 hertz band, it's the nucleus accumbens. Right, this directionality is, again, really important when we develop testable hypotheses, which I'll expand on later. But the really key property here is that I can actually go and say, hey, this is a network related to stress, and I can go to a collaborator that's a neuroscientist and say, this is a network that I found that's related to stress. What do you think of this? Does this make sense to you? Or are there things that don't make sense to you? And I believe that this communication is really important, this back and forth. So in practice, we don't have just one thing going on. We're never going to exactly see only the stress network. We're not going to see exactly only one network. We're going to have lots of things going on at once. The brain does many things at once, right? So for example, right now I am talking and thinking about what I'm going to say next. I'm in a good mood because I get to talk about fun science. I really like all this stuff. And I'm pretty tired because I have 2 young kids at the home in the middle of a pandemic. There's a lot of other things going on in my life that are in my brain right now and are affecting me in different ways. So the model that we're going to be using here is that each one of these brain activities has a separate electome networks. So we have lots of different electrical networks in our brain, and the brain activity at any point in time is going to be the superposition of each brain network multiplied by how strongly it's expressed. So for example, F1 here may represent our happiness network. How happy am I? So when I'm really happy, the strength of that, SW1, is going to be large, right? And it's going to show up a lot in my neural activity. And when I'm not as happy, in that window W, it's going to be much lower. And so really, this is a key model that we're going to be using here. Each latent function has a network that we care about, and we just combine all our activity to get our brain observations. And if we do this, we can actually go back to our problem about different emotional representations. So if we think about this emotional representation here, well, what we can do is we can say, well, each one of these different emotions has a different electome network. And at any point in time, we can actually look at your neural activity and say, based upon the network properties and the data we actually observed, we can estimate how happy you are, how sad you are, how humorful you are. And we can do this by just visualizing each one of these electomes and just mapping the data. So the way we actually do this in practice is by using something called a factor analysis model. So what we're going to do is we're going to simplify data from each window, and we're going to say each window here is YW. YW has C channels with T samples. And each window of data that we're going to use here is a few seconds of data. In our applications, we need to capture things at a relatively fast pace, but not too fast. So we're typically using a window of 1 to 5 seconds. So what we're going to do is we're going to map this to a small number of factors. So we're going to say our observed neural activity is going to be the superposition of SWK. So that is the weight in window W of electome network K, multiplied by dk, which is going to represent our case electome network. So this dk is shared through all of time, and this SWK is a factor score or weight. And we're going to have a model mismatch term as well, but the big idea here is that if we have our observed neural data, if we know what our electomes are, we can actually estimate the vector of weights SW really well. So we can take our big, complicated local field potential data, map it into a small set of numbers that represents what's going on in your brain at that given point in time, which we often refer to as a brain state. Now, the big thing that we really need to talk about is to say that we need to ensure features are interpretable, right? So I've talked a lot about, I want to go from just using machine learning to analyze a problem to using machine learning to develop hypotheses and actually test them. So if we're going to do that, we need to be able to communicate what's going on. And so we have to force that these features are interpretable. So if we use that approach that I gave before, where we said we have this dk and it's just a vector of numbers, if you just do that out of the box, you typically don't get an interpretable structure. So we worked really hard on coming up with approaches where we can actually get interpretability. So one of the things that we require is that our factor scores are non-negative. So we require that there's no negative network that we can observe. And we're going to say that, well, rather than using decay directly, we're going to use something called a latent function factor analysis. So what happens is we define a model that has the properties of the electome network. So we have those spectral properties, and we have those power and phase coherence properties. And then for each window, we're actually going to say we're going to have FWK here, and FWK is now allowed to change slightly per window, but it's going to be forced to share that electome network. So that's actually going to be generated from the electome network. This seems to add a fair amount of complexity here, because it seems like there's a lot more to explain now. And you have to be really careful in the way you do this, but this is an established model, and it's something that we've worked really hard on, both in a 2017 NeurIPS paper, and also we've updated it with a new and improved version in an archive paper last year that should hopefully come out very soon. And in practice, this also seems a little bit complicated to estimate. And if you actually use classical approaches to estimate this, it is a bit challenging to work out all the math and all the gradients and everything. But in practice, actually, you can efficiently approximate this by a deep supervised autoencoder. So we can use deep learning to approximate this system actually quite efficiently. And the code actually is not too bad when you're using modern machine learning frameworks. So I don't really have time to go too deep into exactly how all of this works. I instead really wanted to say, this is what we're doing. Now, how can we actually use this in practice? And I want to take you through a case study. So we're going to look at a case study of social behavior, and we're going to be looking at a two-chamber social interaction test. So we have what I'm showing you here in the top left, we have a freely behaving mouse, and it has two chambers that it can go into. It has a social chamber and the object chamber. And then within the social chamber, there is a friendly mouse, and it can choose to interact with a social friendly mouse. And that's just measured by if it comes within a radius of the friendly mouse, or it can choose to perform an object interaction, which just happens when it becomes within the radius of the object. Now, these mice are, in general, typically prefer social interaction. These are wild type mice, so they're going to typically prefer social interaction. And so we have this question, can we use these machine learning approaches and actually take all this local field potential data that we have and predict whether we are having a social interaction or an object interaction? And the answer is yes. So on the bottom right, we're just showing you that we can get area under the curves way above chance. And we're proud of that, but this is not what makes our method so unique, so I'm not going to actually spend too much time on that. I'm going to focus instead on what exactly makes our model unique and how we're going to use it as a result of this data. And so just real quickly, what we're actually doing here is we're going to use that electome model that I conceptually introduced. We're going to take all of our data, and we're going to map it to a neural network encoder that's going to approximate these latent stores. So we can do this really fast in practice because it's just a neural network to approximate the inverse problem. And one thing that's actually critical is when we're doing this, well, we actually have some information, right? When we're training it, we know whether it was an object interaction or a social interaction. So rather than just training this into what's called an unsupervised objective, we use that unsupervised objective, which is just, can we explain the neural data we observed? And also we require that our latent scores are predictive of behavior. And we encourage that through standard cross-entropy losses that I'm not going to go into too much. So one of the things that's really important for interpretability is not that we use every single electome, right? If I'm saying there's one electome that's going to represent social activity, well, we can limit it to say, we want you to be able to predict the behavior, but only with a single electome. And that's really what we do in practice. So this is the system we're using here. It uses the speed and efficiency of modern deep learning approaches, but it really maintains the electome interpretability. So let's actually look at the electome that came out of this. So we found our one electome network that's actually really good at predicting whether you're in a social interaction or an object interaction. And we're going to test everything now down the line by doing more object and social interaction tests. And we're going to say, well, this is what the electome looks like. And I'm not going to look at every single detail here, but I do want to note there are several frequency bands, but we found that the most dominant frequency band in this electome network was in the 4 to 11 hertz. So in the 4 to 11 hertz, we can look at the drivers, but it looks like the key drivers of this network are the amygdala, the prelimbic cortex, and the infralimbic cortex in the 4 to 11 hertz band. Now, this is our social network, and I'm going to talk a little bit about what we're going to do with it. But before I do that, I just want to show you that this really is capturing relevant behavior. So I'm showing you the scores here as an event-triggered average on our social activity. So when a mouse engages in a social interaction, we can show what's happening with our EN social network activity, or those network scores. So when it engages in social activity, it goes up a lot. And then at the end of an interaction, you can see that you're actually tapering the network before the end of the interaction. And we're showing two networks here. In green is our EN social, that's our social electome network. And then we have an object electome network as well that would signify whether you're in an object interaction or not. So this is really capturing it. There's a pretty wide band here that's standard error over mice because we just don't have that many mice. It's a kind of a wide box, but this is really strong evidence that this network really is capturing social interactions in the data. So now the big question here is this real, right? I've explained a conceptual network model of the brain. It's a pretty complicated model, and there's a lot of data and math that I didn't show you behind it. And I've told you that it can predict whether a mouse is in a social or non-social interaction. So if I were to go to present to neuroscientists, they tend to be a little bit skeptical because they're going to say, well, what evidence do you really have that this is in fact a network related to social activity? And in fact, my collaborator, Kafui Daraza, used to always ask me, is this real? Why should I trust you? And more importantly, how can we test whether this is real or not? These are really super legitimate questions, and they are questions that I think we really need to engage with as computational people. And really, this comes to the point that I was starting my talk with. How can we treat the artificial intelligence or machine learning method as a collaborator to design confirmatory experiments? So let's think about what we want to do. Well, we want this model to generalize. We want this model to be able to decode new animals, right? So if we say get a new animal and we say this is your social network, we want that to actually work on new animals. So we're going to collect new animals and just make sure that it works really well. We want to be able to be linked to interpretable biology across levels of analysis. So we want to see that this holds up across different scales of analysis, right? We're looking at kind of the mesoscale here where we're looking at local field potentials. These are invasive measurements, but they're not individual neurons. So we want to actually see in this context that these electome networks correspond to what's going on in individual neurons, which are considered real drivers of neural activity and behavior in the brain. And third, it should work in new behavioral contexts. And if it starts to do all of these things where we can say it links to interpretable biology, it works in new animals, it works in lots of different contexts, we're going to start getting a lot more evidence that this may be a fundamental principle. So I want to show you a little bit of what we've done on that so far. So first, there's this question of, does this generalize to new mice? Well, because we have trained this neural network encoder, this is a really easy thing to test. We go and collect new mice, we run it to estimate our electome activity scores, and then we just check to see what they're doing. And so we ran this on 28 new mice, and basically the results hold up exactly as you expect them to. I'm not going to say too much more about this, because what I think is a lot more interesting is we really want to know whether individual neurons correspond to our electome models. So as I mentioned in the beginning, when we implant these wires into the brain, we don't just get just electomes and through these local field potentials, we're also able to collect a lot of neural action potentials through a lot of different brain cells. And we get these all over the brain. And so what we did here is we said, well, if we look at all these brain cells, can we actually check whether these brain cells, whether their firing rates are correlated to electome network activity? And so we started by just looking at the social network, and what you find is that 17.5% of all the cells we measured were strongly correlated with EN social activity. So actually, nearly 1 in 5 cells is actually correlated with what's going on in this network. So they are actually linking to networks. And just as a note, I'm just showing you whether a cell corresponds to the social network activity here. not whether it's corresponding to any electome. So if you look at whether a neuron is correlated with any of our electome networks, the number is actually above 50% corrected for multiple comparisons. So these neurons really are strongly correlated with our electome models. Well, and given that we think that these things are actually tied to neural activity, and we have this idea of how we think information is flowing in this electome network, this says that, well, can we actually upregulate this based on a hypothesis that, well, if we have more electome network social activity, so if we increase the activity of that latent network, it's also going to increase your social preference. If you have this network more strongly expressed, you're going to have more desire to be social. So we can do this. Well, what we can say is, well, we know that they're actually related to neurons. This is actually strongly correlated with neurons. So if we had that, well, and we know that it's coming out of these brain regions, in particular, the strongest leader here in our computational models is the prelimbic cortex, we can actually say, go and use optogenetic stimulation techniques where we can take the neurons that start in the prelimbic cortex with terminals in the nucleus accumbens, and we can stimulate them at 10 hertz. And this 10 hertz is chosen based upon the fact that in this electome, we found that this was a very dominant band at the 4 to 11 hertz frame. So we can actually say, well, prelimbic cortex is leading this. It's leading this based on a 10 hertz frequency band. Let's stimulate these neurons at 10 hertz to try to upregulate this network. And so the answer is, what happens when this works? And I'm sorry about this looks to be a rendering error here, but this is saying if we do prelimbic cortex to nucleus accumbens stimulation, the first thing we wanted to do was we wanted to check, does this actually enhance network activation? So if we can estimate our social electome scores, what exactly happens? Well, what happens here is that when we use a control condition, which is a yellow laser that's not designed to respond to the neurons, so it's just the control condition, we see no change in the social network activity scores. And when we use a blue laser that is designed to stimulate these neurons and match with the optogenetic technique, it actually strongly increases the social activity scores. So we're actually upregulating this network as far as we can measure it by using this optogenetic technique. And not only that, we see significant changes in the amount of social behavior that these mice are engaging in. So if we look at whether how much they're choosing to freely interact with the social condition, it's significantly increased under the target condition rather than the control condition. And when we're looking at does it change our object interactions, we really don't get an effect. Now, if you wanted to be a little bit pessimistic here, you could actually say, well, maybe you're just stimulating the right pathways, right? So maybe it's just the fact that you have this first brain region and the second brain region both have been implicated in social behavior before. So maybe we just needed to stimulate that pathway to get this behavioral change. And you're not necessarily using the electome. So to kind of get the question of, well, how much did the electome really matter here? Well, we're going to say we're going to look at 20 hertz and 10 hertz. 10 hertz was in our model and 20 hertz was not. Well, what we find is that 10 hertz across the board, it actually increases the social activity scores, so those electome scores, much more. It's upregulating the network much more. And the behavior is also dramatically differently increased in the social interaction. So when you do 20 hertz, which doesn't show up on our electome, we don't get the same results. So it really does seem to matter that we're using the frequencies and brain regions that we found in this electome. So just to conclude, I have a ton more data that I can show you here. A lot of this data is in a bioarchive preprint. But to really emphasize the key point of today, I strongly believe that we should be using machine learning models as collaborators in experimental design by using data-driven hypotheses. This is what we're working on. And I particularly want to acknowledge the NIH for funding to support this work. I have a brain theory and methods grant that supported much of the algorithm and method development used to generate these results. And my research group is continually working to improve these methods. I really would like to thank Kafui Daraza and the Collective for Psychiatric Neuroengineering, who performed all the experiments here that we used our methods on. And I do want to give particular notes that Austin Talbot and Neil Gallagher both contributed heavily to the computational methods, both recently completed their PhDs and are moving on to other institutions. Just a few key references here, and thank you so much for your attention.